The ancient Egyptian core myth. The least corrupted texts of ancient Egypt are those carved on the pyramid walls of the pharaohs and the coffins of the nobility. Many myths have developed by word of mouth, but the core myth was protected and buried within the tombs and coffins. To determine the pharaonic priesthood core myth, 108 key themes were identified in the least corrupted pyramid texts and coffin texts from 2520 to 1640 BCE. The 108 themes were synthesized into 30 major idea strands, defining textual events and activities of major deities. In this core myth, we meet the dying, rising Osiris, the virgin Isis, the sun gods ferry boat, and the competitive brothers Seth and Horus, the creator god Atum, the scribe Thoth, and the monster serpent Apophis. In the least corrupted funerary texts, microbiological concepts are embedded in a religious narrative, detailing the afterlife adventures of the dead king who desires eternity and transformation. Through this process of recreation, the dead king does not die for millions of years. However, to obtain his desired state in the early cosmos as the firstborn son of the creator god Atum, the dead king must allow specific directions using the earth and stars as guides. By following the advice in the funerary text, the king to be transformed joins the sun god Ra's ferry boat for the voyage through the black duat to overcome the monster serpent Apophis. Isis, the lady of provisions, the scribe Thoth, assists the dead king in his journey to the hidden chambers of Osiris. The dead king judges the two brothers, Horus and Seth, who compete for the throne of their father Osiris. When Seth wins the throne, Osiris dies. When Horus controls the throne, Osiris rises from the dead with the help of Isis, who breathes fire into him and conceives the golden child Horus by virgin birth. The rival brothers are then reunited, resulting in the restoration of the Eye of Horus, along with the birth of the dead king's new form, the golden Horus child, which is nourished by Isis, the rising in the east as a morning star. In the Book of Two Ways, coffin text 1099, the dead king states, I establish millions. With this least corrupted narrative in mind, below are the major signs and their parallels to microbial supermolecules and proteins related to phage lambda's lifestyle and its bacteria host cell as decoded in the Isis thesis. Deflowering the belief in a real virgin. When the Lambda prophet Osiris rises from its dead state, detaches from its protein DNA cross, takes over the replication machinery of the host cell, and clones his own kind, the viral energy source is lactose metabolism, which is now available for the rising virus on the ancient glycolysis fermentation pathway. If the sugar glucose is unavailable and lactose enters the cell, lactose becomes available. Many biologists believe that the presence of lactose has the potential to allow adaptive mutations. The goddess Isis is the sign of the nutrient lactose. And ancient Egyptian texts indicate that Isis is present on the sunball with the dead god or the light. To the ancient Egyptians, Isis represented the Magna Mater, the Lady of Provisions, the great creative power nurturing all living things. According to the text, lactose metabolism is necessary for the birth of viral clones. Genetic cloning may also inspire the creativity of religious artists such as Andrea Mantegna, who conceive a multiple cherubim with a single Madonna, as in the Madonna of the cherubim. Cherubim are celestial beings often represented as winged children, yet in the biblical book of Ezekiel, cherubim are winged hybrid beasts with four faces. The priest Ezekiel's ministry is dated 593 to 563 BCE and marks the transition from pre-exilic Israelite religion to post-exile Judaism. Still, Ezekiel's description for the four living creatures embraces the primary ancient Egyptian signs associated with the pharaoh's chemical transformation. And so we have the connection of Isis with lactose, the Madonna with numerous winged children, clones, and Ezekiel's hybrid beasts that are linked to the major Egyptian signs of divine human, the bull, the eagle or hawk, and the lion. Now consider the idea of Isis and the version of her little Horus child. Christ too was born from a virgin according to Christianity. In ancient Egyptian texts, Isis is a sign of the necessary lactose metabolism necessary for the birth of the Horus child. She is a sign of bacteria, lactose proteins, and is not a real person. The text states Isis wakes pregnant with the seed of her brother Osiris, suggesting that the viral prophet uses the lactose metabolism. This brother/sister union of Osiris and Isis is a sign of viral bacterial sex. Biologists consider the trading of DNA between different organisms as sex, similar to human sexual reproduction involving a male and female who produce a child in the next generation. Human sexual reproduction is called vertical gene transfer. However, bacteria trade genes horizontally within the same generation, brother and sister. In ancient Egypt, the pharaonic family was also practiced royal incest or trading genes horizontally within the same generation. Now, Osiris and Isis are genetic signs, not real people involved in incest. Yet, to explain the metaphor, Isis is pregnant with the seed of Osiris because the DNA of Lambda Prophet is lodged next to the bacteria gene for lactose metabolism on the host cell chromosome. So the seed of Osiris is within her womb. Lactose metabolism gene seats. Remember that when the Lambda Prophet rises and leaves its hidden gene seat on the host cell chromosome, lactose metabolism is provided. Isis is often called the Lady of Provisions in Carpenter's text 246 and the Milk Goddess in Carpenter's text 175. For lactose is the main sugar in milk. Also, her crown consists of a pair of horns between which is a lunar disk, sometimes surmounted by the throne symbol. When the prophet rises through its DNA cross from its dormant state to replicate, it leaves an empty gene seat, that is, an empty throne. Thus, we have a possible correlation between the name of Isis depicting the empty throne or gene seat of the risen prophet and the potential for lactose metabolism. Calling Isis a virgin is one way to express this asexual genetic union related to lactose metabolism and prophet's replication that results in the golden Horus child or viral clone progeny. The ancient glycolysis pathway. For living humans, glycolysis involves the use of the sugar glucose for our cells and metabolic system of breathing oxygen for energy. However, bacteria can derive energy in other ways, such as fermentation, eating nitrogen gas right out of the air, precipitating iron and manganese, or combusting hydrogen with oxygen to make water. Microbes invented fermentation, and humans have turned fermentation into a technology producing bread, cheese, wine, and beer. Human fermentation technology may be another sign of the ancient glycolysis fermentation pathway, a nature inspired design invented by microbes. Perhaps our evolutionary predilection for eating sweets is also related to this useful metabolic pathway that is with its message of survival via various sugars. As we shall see, our microscopic behavior often points to dynamics on the microscopic level. Biologists believe that before the advent of oxygen, the ancient glycolysis fermentation pathway was the source of energy production for earlier organisms such as bacteria. This ancient gene expression network can function without oxygen or glucose by using lactose, the predominant sugar in milk. An outer membrane protein door known as the lamporin is the gateway to the ancient glycolysis gene expression network. The channel protein LAM also the passage of sugars, especially carbohydrates such as maltose, maltodextrins, and glucose. Lamb contributes 70% of the total glucose import capacity of the cell. Lamb is also known as the gateway to the maltose transport system that the bacteria virus lambda uses. Conveniently, phage lambda enters the bacteria cell at this receptor site or gateway to the ancient glycolysis gene expression network, a surprising coincidence. It then travels along the bacteria chromosome to the lactose genes, where it integrates the chromosome by a site-specific recombination process called a Holiday junction. So make sure we see figure 1.3. The virus is now a prophage, but it has the potential to take over the replication machinery of the whole cell, using it as a factory to clone its own kind. This mass production technique is the lifestyle of lysis, or the lytic lifestyle, and it has been in existence for billions of years to clone viral heads and tails, long before Henry Ford picked up the same production technique for cloning factory automotive parts. In ancient Egyptian texts, the sunbark or light damages the cell, activating the cell's SOS response. At least 42 genes that function in DNA repair, recombination, mutation, translesion, DNA synthesis, and prevention of the normal cell cycle are de-repressed. These genes are usually repressed by the cell's monster LEX-A repressor protein. However, the SOS response activates the multifunctional enzyme REC-A that slays the monster LEX-A protein, so mutation can begin to fix the damage. Numerous Egyptian textual references also emphasize the importance of cultivating or brewing barley. The sugar maltose is used in the brewing of barley, and Osiris, sign of the lambda prophet, is closely connected to the harvest of barley in ancient Egypt. So the brewing of barley is another sign of the maltose transport system in the ancient glycolysis fermentation gene expression network used by phage lambda. This same system also allows the transport of lactose into the cell by a low affinity pathway. Thus, a variety of metabolic sugar mechanisms, glucose, lactose, and maltose, are available in the ancient glycolysis fermentation gene expression network, showing how living organisms are efficiently organized for survival. The second death, gene regulatory networks control gene expression relative to environmental changes. The ancient Egyptian text explained the deceased person's DNA can transform and survive via the ancient glycolysis fermentation gene expression network, and is also known as the maltose transport system used by phage lambda. The ancient text explained that an awareness of the signs is necessary for survival. The text also tell us that that those without knowledge are deliberately and systematically destroyed in a protein degradation manner similar to human genocide. Survival or dissipation of waste slash heat is simply the difference between knowing the signs or not. Thus, ancient Egyptian literature, artwork, ritual, and architecture model a complex viral replication cycle for human DNA survival at a death transition along the ancient glycolysis pathway that runs with or without oxygen. The Egyptian text explained that without knowledge of this ancient sugary gene expression network and its land pouring protein gateway for phage lambda, one meets death a second time in the microscopic domain that we all return to when we quit breathing and die. So the secret Egyptian texts inform us that our cells have the potential for survival as well as a role for phage lambda to play relative to human survival of DNA at death transition.